Perot, you can take it away.
All right, let's fingers crossed this share screen works.
Okay.
Which version do you guys see?
We're in your view, so you want to flip it to yeah.
Better?
I think it's shrunk.
Good.
Is that good? Okay, that's just my
Oh,
view.
I think, no, it is shrunk.
Yeah,
It's not,
jump
it's feeling,
jump back and use presenter view. I think that
no, that's weird.
is that as big as it gets?
So swapping, that doesn't do that, right?
Yeah,
No.
try use slideshow and up at the in the top left there.
Oh, now.
Yeah, I think that's what you want.
Okay, cool. All right. Sorry, I always got to test it 10 more times before I actually get this off. So thank you, Dana, for introducing me. And so, yeah, today my talk is bite-sized genomics, decoding tick population genetics, acaricide resistance, and beyond. I also snuck in something that isn't tick. So my work oftentimes includes a lot of different species. And so ticks is being one of the main ones, but I also have some secret other fly species I work on. And so to help fill up the time, I filled that in, but hopefully it's interesting, and so I'll include that. And so I think Dana did a great job of introducing me, but I know that there's a lot of students in here. And so I wanted to kind of just reinforce where I came from. And, I was recently got my PhD from UC Davis, and I did my postdoc with the USDA, not knowing that like government research was like a thing that people could do. Like I thought that if you wanted to be into science, you go directly into either only academia, or maybe you come into a company. So I always like to highlight that I managed to go from my PhD into like a government research unit and get some experience that way. And so the government has been an amazing place to grow and develop my science, story. And so for those of you that are considering your career, I always like to, pitch that if I could do it, a lot of people can do it. And that, there's a bunch of really neat opportunities within the USDA ARS. So and so as he mentioned, I'm part of the veterinary pest genetics research unit. This is just something I copy and pasted from ChatGPT to see if I knew what I did as a unit. And so, but really, we're an initiative from the U.S. Department of Agriculture, and we focus on leveraging genomic and big data solutions to kind of evaluate the risks posed by arthropod vectors to develop practical solutions for U.S. farmers. I think my 5-year-old sums it up best in that I just smash bad bugs and get information out of them. And so he's been my biggest cheerleader. And I think, oddly enough, I've given a talk to his school, and that went just as well as the talk went with the high schoolers. So, 5-year-olds ask great questions. And so currently we're going through some really good, big renovations. So the unit is this picture is outdated. We have now a brand new facility that we're hoping to open in January. But the reason I mentioned this is that we have some amazing capabilities at our unit where we're highly involved in sequencing and doing big data generation. So some of the things we have are PacBio sequencers, the Sequel2E and the Revio. For any of those people that are knowledgeable about this, this is for long read sequencing technology. So we can use these platforms to do things like genome assembly, annotation, long read microbiome studies. We also have some smaller capabilities in the short read sequencing space, which is like Illumina. And so stuff like transcriptome sequencing, SNP identification, and resequencing. We also dip into the nanopore space, which I think fills the same niche as the PacBio. So this is another like tabletop sequencer that allows you to do like long read sequencing. Also gets you some methylation information. So, but I wanted to highlight some of the resources our unit has for those of you that might be considering internships or also future collaborations. I'm always here to pitch ideas with other faculty to give you an idea of what we have. And so, my talk is going to kind of cover a little bit of like a sampler platter across of different projects I'm working on. One of the main projects is being this cattle fever tick. So, and then I have some work going on with a collaborator on Amblyoma americanum. And then finally, I swapped out ticks with a fly and the New World screwworm because this is becoming a very big deal recently, where it's starting to become an emergency response that the US is having to have with this New World screwworm that was a species of pest fly that was previously eradicated through the US and hopefully will continue to stay so. So, I'm a geneticist by training and not a very good entomologist, so I always have to remind myself what a cattle fever tick is. And so Rhipicephalus microplus is the southern cattle tick. And this was a tick species that was brought over into the US and through Mexico, likely through horses from the colonization period. And cattle fever tick is a single host tick. So the ticks will go, starting from adulthood, they'll go in and get engorged off of cattle or whatever. Then they'll lay their eggs going through the larval and nymph stages, and then essentially can come back up and then, spread back out into adults. And their life cycle is about 9 months. That depends largely on humidity and temperature. They like, I think, high humidity and warmer-ish temps, not too cold. But cattle fever ticks are especially concerning because they potentially are a vector for Babesia bovis, which the ticks act as a vector where they can pass this pathogen into ruminant hosts, and they can cause some nasty things, as you can see here on cattle. And so back in the early 1900s, there was an eradication program that the U.S. Department of Agriculture took part in, where essentially over 700,000 square miles of the U.S. were identified and attempted to isolate down. So the ticks were actually eradicated over time, all the way down to the southern U.S.-Mexico border. And so the program was very successful. We managed to essentially wipe out any outbreaks of this tick as far as the Deep South. And so my unit is in Kerrville, Texas, as you mentioned. And so at any point, we were looking for an excuse to be cowboys. So this seemed like the perfect opportunity for the USDA to deploy these tick-mounted inspectors. And so you'll actually have cowboys essentially going down along the US-Mexico border, looking for stray cattle to kind of round up and enhance our surveillance abilities, to make sure that, once again, no cattle are crossing through the US-Mexico border or wildlife that may be harboring some of these nasty species. And so we have a lot of challenges that come up with controlling these cattle. So right now, one of the main ways is pesticide treatment. So I recently just got back from a trip where I got to interact with a lot of cattle. And one of the coolest things interesting things, I should say, is how cattle crossing from the Mexico border come into the US. And so, we've developed these devices where these are like gigantic trailers where the cows will kind of walk through this shower of pesticides. So oftentimes they'll be sprayed with like a Kumafos spray, or they go through this like assembly line method where they jump off this platform and do like a nosedive into this, we're going to call it a water bath, and come out, and the ticks will actually get washed off of these things because they're essentially being treated externally with these this compound treatment. So, that's great, but I'm sure, and in the future, I would love to help develop solutions where we're not just relying only on systemic dipping as a mechanism for control. And so some of the things is, clearly a cow nose diving into a pool, it can cause injury, and there's often a very expensive time-consuming process for this. And in addition, one of the worst things you can imagine is that Acaricide resistance is something that comes up. And so our lab has been working on studying Acaricide resistance, where we're finding that populations in the Mexico side are developing resistance to specific compounds. And so I'll have more about the data on that. But not just cattle, but it turns out wildlife are also vector or act as transports for these vectors. So we have things like the white-tailed deer who eat all my plants in my garden, to red deer. I don't think we have much moose in Texas, but meese. But they are a problem. And so nilgai also are potential harborers for ticks. And so one of the biggest challenges we've been actually facing lately is the increase of native and exotic ungulates. So their populations have exploded, and one of the worst culprits is this guy here. This is a Neil guy who is very pretty, but it turns out that they were not properly brought over from, I believe they're African in origin, brought over to the US, and they ended up being a amazing host for many of these ticks. They have a large range, they can graze. They don't actually like to settle in a location, they like to go all over the place. And one problem is it seems like a lot of ranchers like to harbor them. So turns out wild game hunting is a big deal. And so, and these guys don't seem to respect the US-Mexico border. And so that's a troubling thing where people will harbor these organisms, but at the same time, they are a terrible vector for disease, potential vector for diseases. So while we have treatments for wildlife for deer, such as like using corn feeders to administer treatment against against these ticks, we don't actually have the same solutions for nilgai. And so some of the things we're going to have to do is enhance our ability to do wildlife surveillance. So, enhance our ability to monitor these things from afar and actually get better ideas of where these things are located. And so that's one of the biggest challenges. So a lot of what I've been talking about is just about these things on the Mexico-Texas side. And, you guys all comfortable in New York may not care about this, but, economically, they can be disastrous. So we all care about money. But at the same time, there's potential for these things to make it up north. And so there have been at least some confirmed affected sites up north of Texas. Now, once again, they're not in the US. So these things are constantly being eradicated and removed. And so MicroPlus has a genome that was sequenced by a group in China, Zhao et al. In 2020. And so they did an amazing job where they sequenced a bunch of different tick species, and they found, were able to get down to nearly a chromosome level of an assembly. So when we look at their genome assembly, they've identified putatively 11 chromosomes. But when if you look at histochemical staining, you find that, there are 10 chromosomes and a sex chromosome. Now, in the bioinformatic chromosome that the group came up with, we can't seem to find the X chromosome. It seems like, we would expect something about 528 megabases, and we don't see that in the assembly by that group. And so that suggests that either there's some issue with their assembly or that they were just not able to sift through the data and find it. But the X chromosome should be this large piece of the genome that seems to have gone missing. So once again, the bioinformatic assembly does not seem to match what was previously and historically found through histochemistry. And so the challenge and what our goal was, my unit's goal was at the time, is to identify, can we find the sex chromosome? And so, as I mentioned, the current assembly lacks the putative sex chromosome, may have been due to a misassembly. And then the question is, can we computationally find a way to correct and assemble the chromosome? And so single nucleotide polymorphism density mapping is a method that we can apply here. So SNPs have a uniform pattern across the chromosome that can be used to determine the rates of recombination in sex chromosomes. Now, this density of the SNPs, so what we did is we mapped the density of these SNPs. And what you would expect here is that there should be a pattern for every chromosome where you would have a ratio of SNPs, and then you would see this kind of falling off, hinting at the acrocentric chromosome structure that we would expect. And so we see that structure except in chromosomes 4 and 8. So what we see here is we actually see two tails in 4 and 8. We see a drop here and then a drop here. And then chromosome 10 has this drop here, but we don't see that drop anywhere on chromosome 8. Now, the hemizygous nature of the X chromosome would have less recombination events, resulting in lower number of SNPs. And so, trying to small that. Okay. And so what we want to see is the differences among the chromosomes. We would like to 5 different groups, right? Chromosomes 2, 8, and 9 display a significantly lower percentage of SNPs per nucleotide, similar to the hemizygous sex chromosome. So chromosomes 2 and 8 are outliers in the ratio of in the percentages of SNPs compared to the other groups. So once again, that kind of hints as so sorry, and I think I should highlight this a little bit more. So these 2 chromosomes kind of standing out really potentially hints that there's something wrong with this chromosomes 2 and 8 rather than the rest of these. So chromosomes 1 through 11 keep a pretty typical ratio of SNPs that we would expect. And so perhaps there's something weird with these 2 chromosomes, hinting that they might be candidates for either being 2 separate parts of a chromosome that need to be fused to one another, or that they were just misassembled and that those need to be corrected. And so to further identify if this is the issue and if there's a potential misassembly with chromosomes 2 and 8, we tried to use copy number variation of select genes that are associated with sex-specific, that has been hinted as sex-specific activity. So things like double sex, transformer, are genes that are known to drive sexual variation, being either male or female. And so what we would expect is a gene expression ratio of half, right? Because it's a hemizygous, so you would expect one rate of half the level of expression of a gene in an X chromosome in an X species. And so what we actually see is that there's 2 genes that actually far exceed this, is this double sex and TRAP2I3. And so, but the rest of these genes all keep this like hemizygous nature where we see, half the level of expression. So what that means is that, if we look at these genes, so on 2 and 8, we suspect that half of that is being expressed. So that would be something that would be potentially double dosage compensated as an X chromosome. So if you have 2 X's, you would have double the expression. And so I think that kind of highlights that potentially the chromosomes 2 and 8 and those portions of 2 and 8 are actually misassembled. And so what we can do then is take chromosomes 2, 8, and potentially 4, cut them and paste them back to correct for the genome misassembly. And so what we believe we have done is that we've identified that there are genes on 2 and 8 that are biased, that are representing a ratio that indicates that it's a potential X chromosome. We can stitch those chromosomes back together, and what we end up seeing is that we get the X chromosome of the expected base, of the expected genome size. And then now we reduce down the number of chromosomes back to the appropriate numbers. So now we have 10 chromosomes with the X chromosome identified. And so the chromosome, the assembly on NCBI, we have some statistics here that would, in addition to the fact that we corrected the chromosome structures, we also found that our completeness and our overall statistics are improved. So we're able to get a more complete genome, just essentially saying that we got a better genome, and that overall we managed to reduce the amount of missing information in these data sets. So now what are some potential applications with these improved genomes? So earlier I kind of hinted at the geographic distribution of acaricide resistance. And so something that we wanted to look at is, can we now, with this improved chromosome level assembly, identify genes that are associated, or look at patterns of gene expression associated with acaricide resistance? And so we, some of the things we're doing are selecting for high permethrin resistant locations. So you can, some of the names of these are useful, but for the most part, you need to know that we, and some of our, in the study, we have permethrin resistant strains. We also have permethrin susceptible strains, which is this Deutsch strain is like the lab strain. And then we also look at like wild caught resistant samples. And so what we can do is, like, looking at the patterns of gene expression, we end up seeing that there is, some sort of correlation with this is susceptible group and this is the resistant group, and it mimics, what we see here. So essentially what we're saying is that, looking at an entire set of gene expression data, what we can find is that, patterns begin to emerge among the resistance group and the susceptible group. So to kind of pull this further, we're looking at other types of compounds too. So for those of you that are unfamiliar, fipronil, amitraz, ivermectin, and coumaphos are all traditional acaricides that we use to treat against ticks. And so what we can do is look and see how many genes are differentially expressed by locations that have a high resistance, so high, or a low resistance. And so when we look at within location, when we look at a laboratory strain that's known to be susceptible to the fipronil, and we compare it to a strain that is known to be resistant, we see, a large number of genes. And really what I wanted to kind of emphasize in these plots here is showing that, likely this is more genetics. So there seems to have been some sort of adaptation within this location of like L. zamora, where if you took the L. zamora that was selected to be susceptible and then selected to be resistant, you find like a set of genes that are likely to explain resistance to the acaricide in L. zamora. But when you look at a susceptible genomic strain against a resistant location strain, you see a huge amount of genetics. So likely a lot of these genes here are just location specific adaptations to being a L. zamora tick. But at the same time, you do have, candidates you can whittle down to finding that might be explaining the that might be explaining the specific resistance to acaricide resistance. You see that a little less so with amitraz. It seems like amitraz, you have a huge number of differences between a resistant versus susceptible strain. And despite the location, you still have a huge amount. But I think it really is interesting that, based off your genetics, you can see where adaptation both to the location and to your specific compound is kind of coming up. And so we're trying to dig into like what those genes are. So, we have candidate genes where we find that the expression is high in the high resistant group, and the expression is low in susceptible groups. And since many of you may not know this, with new genomes, you don't actually know what these genes are. And so right now we're digging into like what these putative genes are and what they might be to kind of better understand their function. And kind of lastly, off of the gene expression trait, one of the last things we can do is this whole genome co-expression network analysis. So what we can do is we can take all of the data from all those locations and all the comparisons with gene treatment and identify if there are nodules of genes that are co-expressed, meaning that like, are there patterns of gene expression that follow, based off of treatment, based off location. And so what these color modules here indicate is that, the darker the more intense of the color, the more likely you're seeing a cluster of genes that are, associated with, San Ramon low resistance or susceptibleness. Another cluster of genes that are for example, these two comparisons, a resistant strain and a susceptible strain. Blue, this module of sets of genes for blue is likely explaining high resistance to coumaphos and San Ramon, but you don't see that pattern in any other location. And so what we think here is that this data will be useful for is kind of really isolating down what those genes are that are specific to the location and the potential compound that they're associated with resistance or susceptibility to. So, another good candidate here is these two, this nodule here, gray, is likely going to explain what is happening in San Alfonso. So in addition to gene expression, something else that we're kind of working on is microRNA technology. And so microRNAs are these small fragments of like 18 to 22 nucleotides that can regulate gene expression. They also can help regulate the host expression too. So we found that in addition to regulating your own gene expression, ticks have figured out a way to use extracellular vesicles, which are these compounds emitted through the salivary gland, and they actually will package potentially portions of DNA or RNA that can help delay in wound healing and enhance the tick's ability to feed. And so there's been a lot of studies in understanding what are the microRNAs, what are they doing, and how are they actually able to do things like delay wound healing and enable the tick to continue to feed. And so some of our early studies are going in and looking into what are something like in Ixodes scapularis. And if we look into Ixodes scapularis and look at extracellular vesicles and what microRNAs are being expressed, we get these kind of profiles here within the salivary gland and within the extracellular vesicles. So MIR10 is a candidate, and I'll go into more detail about what those are. But essentially, what we wanted to do here is just look at, for a quote unquote typical tick. So Ixodes scapularis is just your average run of the mill tick that gives you Lyme disease. We're able to at least identify ticks and what the feeding behavior of Ixodes scapularis is. And now that we have the microplus genome, we're able to do something very similar where we're able to profile the microplus salivary gland microRNAs and what's inside the extracellular vesicles. And I think the thing that we found that was really interesting is this MIR10, MIR100 are a significant portion of what's inside of these extracellular vesicles and salivary gland compounds. And so MIR10, MIR100 is a microRNA that's been known to be a precursor for wound healing. So, I think it's really interesting that, now that we have an improved genome, we're able to hone in on what the microRNAs are and their relative abundance within the mechanisms or processes that they're using to feed on hosts. And so I think that that's a really cool example that we're finding that, okay, so this is a really great target, and now we can start to hone in things to potentially dysregulate MIR10, MIR100 within the tick feeding process. And so hopefully in the future, we can start to maybe approach things like developing vaccine targets to help as a treatment option. And then something that we're also furthering is novel identification. So those are microRNAs that are known. And so also looking for new motifs for, de novo microRNAs that might be either species specific. So these are Ixodes scapularis. And so we're finding, an interesting profile within Ixodes scapularis for novel microRNAs. And something kind of weird is that it turns out really for novel microRNAs, so unknown things, the microRNAs that are in the extracellular vesicles are predominantly made up of just this one microRNA. So we're not sure what that is. Why is Rhipicephalus microplus essentially pumping a extracellular vesicle solely with this single microRNA? So, and it doesn't come up in the salivary gland. So it's an EV specific microRNA. So that's kind of interesting. And so a little summary for that is that we hopefully fixed a misassembly that we believe and have identified the putative sex chromosome. We can do things like genotype associated with resistance to acaricides, and hopefully determine things like genome-wide SNP frequencies. That's kind of a future direction. And that we also demonstrate that microRNAs identified from the genome can be putative targets for tick control. And so now I'm kind of roll into another portion of the tick talk where it's more about population genetics. And so once again, I've been playing with a lot of ChatGPT. And so I was like, give me the most Texas-y Lone Star tick you can. And then after like the third iteration, you get this tick. Don't ask me what iterations 4, 5, and 6 look like. It almost gets obscene. But so Amblyomma americanum is a really interesting tick. And ticks in general have this really weird, interesting thing where, so let's see, humans should be here, which are about, the genome size of a human is about 3.2 gigabases. Chickens are about 1, fruit flies and other things are about 300. So they should be somewhere around here. Aha. And so ticks actually have genome sizes very comparable to humans, which is pretty odd, right? Once again, ticks many of them are probably the size of a pen tip, if not a little bit bigger. And these things have gigantic genomes that are highly repetitive, very essentially complex. And so that's kind of different than what you would expect in the tree of life in terms of usually the bigger-ish you are, the more complicated your genome. And so that creates this issue of like your genome is large, you have to have, novel technology. And so referring back to some of our systems, we have, sequencing technologies now that are going to help us improve our continuity with a greater completeness for enhancing resolution of repetitive and complex regions. So what we have now is technology that allows us to power through and sequence longer bits. So thinking about assembling a book now, instead of working with single words and trying to assemble those back, now we're having pages of information that we can kind of paste together. So something that I don't think I talked enough about is that the genome of Embryoma mecanum has been published, and it is a pretty decent genome. Unfortunately, so N50 is a statistic that lets you know roughly how complete it is. So, what are the largest size fragments that you have? And the one that's currently published actually does not have a great N50, meaning that there are a lot of small fragments found throughout that assembly. We are able to get that, 10 to 100 times better, but it's still not to the level that we're able to get to things like chromosome level assembly. So we're not able to say that here, ticks 1 through 10 chromosomes are found, but at least we're not into the thousands of pieces. But essentially what I'm telling you here is that we're at least improving the draft genome that's currently up on NCBI, hoping to get to the point where we have a strain that comes from our lab and a strain that comes from Texas A&M, and that we're trying to get this genome to be as great of a quality as possible. And so, to give you a peek under the hood and what that really means, so this y-axis is our genome, and the x-axis is what you can find on NCBI. And so you can see that, for scaffold 2, we're at least able to place a bunch of these putative contigs that the previous authors were able to assemble, and we can kind of paste it together to show that, okay, actually that's all of our second scaffolds. So we're getting really close. This is, I think, really highlighting how really complete our genome is compared to what's up there, and that we're nearly to the point where we can call some of these chromosomes. So we also detect that some of these things are misoriented. So you see forward and reverse orientation. And so we're currently working on making sure that we get the proper orientations for a lot of those scaffolds to, once again, hone down the completeness. And so with the chromosome, we want information like the genes, right? Because the genes are going to give us gene expression information, and that's really going to be what a lot of people, scientists in particular, are really interested in doing. And so what we get are a large number of genes. You would expect roughly 20,000 genes, but when we lift over information and look at what genes are reported, we get roughly 28,000 genes, which is quite a bit more than you would expect. So I think that's all to say that at least we have genes and we can place some of them despite the fragmented nature of our genome. And so what we can do with this is, what we want to do is track the distribution of and range of Embryoma americanum. And so, as of today, I believe that the range of this tick is pretty broad. And so for those of you that don't know, Embryoma americanum can cause the meat allergy. I believe it's one of the potential ticks that when they bite you, you can develop a meat allergy, which is more of like your autoimmune response rather than like a thing that they're passing to you. So to continue to detect this range, we are working with a collaborator from Texas A&M, Oklahoma, and Arkansas, where they're sampling ticks from these various locations. And can we see what local adaptation and population genetic forces are happening in these regions, and how they're working to expand and develop and distribute. And so for those of you that maybe are aware of this, there's often this issue of reference bias. And so at the moment, we currently have those 3 versions of our genome. And so we actually get different average number of SNPs called based on the kind of reference genome you use. So I think I just wanted to kind of be candid with this, in that, based off of what genome reference you use, when you look at SNPs, you actually get different numbers. And so I think that that's really interesting. That at least we're able to call more SNPs, probably likely because of the more completeness of our genome. And so So this here is a principal component analysis of those different locations that I previously mentioned. And so what we're finding is clearly they seem to be segmenting and distributing based off of like the state. So you can kind of distinguish like, okay, here's what a Texas tick looks like. Here's roughly what an Oklahoma tick looks like. Don't blame me. I didn't learn geography, so I know that AK is not Arkansas, but it's Arkansas. And so we see distributions in that generally within this principal component analysis that these ticks are actually pretty unique. So they're developing adaptations to their state or to the like roughly the locations that they originated from. So, and one of the ways we can kind of quantifiably measure that is FST. So FST is a measure of population variation. So, you know, and to give you some scale, 0.15 is the FST difference you'd see from a wolf to a coyote. And then within human and sub-Saharan African populations, you actually see the same thing. So you could say that like a European population to an African population is about the same difference in FST as a wolf to a coyote. I don't know what, anyways. And so when we look at FST differences across these populations, what we ended up finding is that, you know, some of these populations show really high levels of differentiation. So 0.03 would be considered high. So Arkansas seems a pretty distinct, unique location. Whereas things like, you know, Lake Stanley, which is a location in, I believe, Texas. Oh, I think I colored it. So these are Texas, these are Oklahoma. And so really, I think this is like a quantitative measure of just showing that like the blue group is sticking roughly with the blue group, and they're really similar to things that are to blue, and they're very distinct from things that are not, you know, the same color. So Arkansas is really different than all the other groups. And so this is more of just a quantifiable measure for that. And lastly, for the Amblio americanum, assemblies can actually be confounded by the presence of contaminants. And so for Amblio americanum, there's been this known endosymbiont. And so when we looked at our genomes and actually we look into the data, what we ended up finding is that we see the presence of two really well-known endosymbionts, Ehrlichia and Anaplasma. And so we find that there's actually variation in the relative abundance and how much of those endosymbionts are present in those locations. So each of those locations has the same number of individuals, but we're seeing different abundances of these endosymbionts. So that kind of leaves two interesting questions, which is, you know, can you use this information of like amount of endosymbiont present in a population to track the population, and why would different populations have different levels of endosymbionts, contaminants. So I shouldn't call them contaminants. Oftentimes it's like stuff inside your organism. So whether or not that's contaminants. So lastly, Lone Star ticks are difficult to assemble, and we're getting better. You know, populations show that, you know, there is local adaptation. So these ticks are at least differentiating by location. And there's something interesting with being able to detect microbial contaminants, quote unquote, that's prevalent among all the species. And so something we want to do here is enhance our ability to detect more things inside the tick and see, you know, if there is presence of nasty stuff or if there's, you know, ways we can track that. All right. Lastly, I'm going to talk about New World screwworm and just some fly. And so what I wanted to do here is, this is something that's becoming more and more important as this issue is growing. And I wanted to kind of highlight it because our lab is very involved with some of the population genetics and sequencing efforts that's going on here. So the New World screwworm was a major pest of livestock and animals. And it actually made its way all the way through most of the south of the US and all through Central America, and is still present in South America. Now, it was eradicated from the north using the sterile insect technique, which is a method where you can irradiate males and essentially do these mass releases of these sterilized irradiated males. When they mate with females, they no longer release viable offspring. And so it was a big issue in the past, and over the time, our lab actually is coined after the person who developed this technique that used sterile insect technique to drive down the infestation all the way down where it ended up but we eradicated them all the way down to Panama. And so we created a barrier zone at Panama. And so essentially now we release, the USDA and APHIS release 20 to 50 million sterile flies throughout Panama every week. So I don't
have
think
a
you guys
reminder
truly
on
can.
your screen, Perot. I'm sorry to cut in.
Oh, okay.
It's
Good that it
covered
says
up, like
Rutgers.
there's a reminder that the pop-up that covered up like in a, yeah, there you go.
Okay, there you go.
Sorry.
No, it's okay. So yeah, and so this is a big initiative where we're rearing flies to sterilize and release to control this population of screwworm to prevent it from making its way up north. And I think the previous figures had demonstrated that in 2016 we've actually had an outbreak in the U.S. in Florida, where we found that around 20% of the endangered deer population died only after a small outbreak had occurred and was re-eradicated after only 6 months. So once again, this initiative has been great in controlling and preventing it. Now, unfortunately, as of 2023, and I think if you talk to anyone that's involved in this project, they lose sleep at night because the cases are starting to go up, and that what you're seeing here is distributions of breakouts across from 2023. And so you're seeing more and more cases breaking up. And so you're seeing things making its way up through Costa Rica, and potentially, last I heard, is Nicaragua. And so, I'd like to say that the USDA has capabilities to continue to breed this, but a plane can only fly so far, and we can only breed so many flies. And so, this is a really interesting issue where now we're seeing this issue kind of come up again, and that the sterile insect may be faltering to some extent, not because of what it is, but perhaps strain variation. So, in order to present and prepare us to better respond to this, we need to be able to identify the genetics of our strains and of outbreak strains to better deduce where they come from and how they're distributing. And so work in my unit and with collaborators, Mackenzie Tejan, we're looking at, seeing if we can distinguish between the location, the, Central American and Caribbean islands, and see if we can develop sort of like fingerprint of those locations. And so when we did sequence some of these populations, we were actually able to develop a network analysis that allows us to say, okay, well, based off of similarities genetically, we found that like that Florida strain actually is more associated with this inner Caribbean location, or rather than this outer Caribbean location or South America, thus kind of indicating that this may have been the likely source of that Florida outbreak strain. And so once again, yeah, it kind of just determined that the Florida outbreak was out from the outer Caribbean. Okay, outer Caribbean locations. And so in the future, we're hoping to use whole genome sequencing to kind of improve the reference database and thus enhance our ability to do more strain development and enhance the genetics of what we know of those species. In addition, we're trying to find out, are there ways we can look at like evolution and the history of these species, of these blowflies, to maybe find control methods? And so screwworms are these Cochleomya. Hominavorax is the New World screwworm. And we're currently working on developing genomes for Minima and Macellaria. So something that we're kind of looking into a little bit more. So Hominavorax is an obligate parasite, meaning it feeds on something living. And Sarcophagus is only feeding on dead stuff. So within that clade, you saw an evolution where things were originally feeding on either only living, but in this clade, you had three separate different events that came up. One that only wanted to live off of dead stuff, one that wanted to live on either dead or alive stuff, and stuff that only wanted to live on dead things. So this is kind of a perfect example of a study that where we can look into seeing the evolution of some of these behaviors through this kind of genome sequencing. And so the Hominavorax genome is available and up, and we're trying to work on improving it. And so, but we're currently working on developing sequencing efforts to develop genomes for the Macellaria, which is another XY system. And so to do this, we're trying a trio binning approach. So for those of you that are unfamiliar, you can sequence a strain from the mom. If you sequence the dad and you also sequence the offspring, you can deduce which parts of the genome came from who, and that helps you resolve down the haplotypes by doing this. And so what we're finding, okay, yes, and then you get the reference assembly. And so what we find is that when we do this, we actually get a very high quality genome assembly. And so we get, a really good haplotype for the mom or dad, and then the whole genome itself. So something as little as 30x coverage or as high as 30x coverage allows you to get something that's really, potentially really scaffolded well, meaning there's very few fragments. You have a really high single copy number and very few duplicates and missing pieces. And so we find, that we're able to at least identify the autosomes, so chromosomes 1 through 5, and a putative sex chromosome X, and then maybe the Y. So if we look at, reads that go from mom and reads from dad, we don't see any here, but we see all these reads from dad, likely indicating that this one is a potential X or Y chromosome. And so I put this up, but this is more just showing that like the genome looks pretty decent compared to the hominivorax genome. So this is the screwworm, and our genome actually looks pretty similar to some extent, if not equal or better quality. And so, as I mentioned, we're going to start to do some synteny analysis or chromosome or species comparisons, where now we can start to see, okay, we can track, what parts of the genome from other species line up with with the genome that we made in addition, and see, where has evolution kind of gone, and has pieces of the DNA moved on across clades. And so what that could look like is, studying something like odorant receptors. So something where we can look at, the expansion or contraction of these gene sets. So odorant receptors are really important with what one would think discovering rotten or dead meat. And so we can see, how many of those genes are conserved across Macellaria and how many of them have come across hominivorax. So perhaps to smell something living, you don't need as many odorant receptors as you need for Macellaria, which needs to have the ability to detect, dead and dying things. And so overall, we've just kind of showed that we are able to do better genomes with fly species because they're not as hard. And that, hopefully we can do some things like more evolution studies to get a better idea of like, what might be happening evolutionarily to drive behaviors or traits of interest. And help us with some of these like issues that may be coming up with strain evolution. And so that's kind of so with that, I just want to say thank you to all my collaborators and students. The work in the cattle fever tick was largely driven by a student, Jason Tidwell. My work with Amblyoma americanum is with Pete Teal down at Texas A&M. Adela is also a person who has been working with me on the microRNAs, and the fly work is all done by Paul Hickner and Mackenzie Tejan. And once again, did I have this? Oh, well, there's some positions with the ARS. I know that, if any graduate students are near graduation or postdocs, there's opportunities. And so these might be out of date, and I recommend you to check out USA Jobs. And for graduate students going on, GS11. For undergraduates, anything around 6 to 9 GS level, that's just like a pay grade. So with that, I'll happily